Hello. Today I will be giving a review about the paper written by a group from the Department of Electrical and Computer Engineering in Boston University entitled Label-Free Microarray Imaging for Direct Detection of DNA Hybridization and Single Nucleotide Mismatches. So, a brief background. The DNA microarray is a technique that displays a grid of DNA segments of known sequences and is used to test and map DNA fragments. This technique is utilized in expression profiling, cancer research, and medical diagnostics applications. There are immobilized known DNA fragments on a substrate-applied glass sample arranged into spots that contain several hundreds or thousands of these segments. Expressed genes from an experiment are applied to these fragments because hybridization of different DNA fragments to the expressed genes is used for DNA detection and identification. Another application of DNA microarray uses fluorescence tagging. DNA fragments extracted from a specimen are labeled green, while reference standard samples are labeled red. Then the two types of fragments are hybridized to a DNA microarray, washed and read via a scanner. The ratio of red to green signals after hybridization is indicative of which genes are expressed and their expression levels in the sample. These techniques are very important when identifying DNA sequences that cause certain diseases and determining how long it may take to respond to treatment. Previous research has shown that variations in DNA by single nucleotide polymorphisms, or SNPs, are usually the cause of specific diseases, and the detection of SNPs could be useful in diagnostic purposes and treatment predictions. However, detecting SNPs using fluorescence results in some complications. Distinguishing single mismatched hybrids from perfect DNA hybrids require the technology's ability to detect different binding energies. Conventional fluorescence is unable to relate the intensity to binding energies, making it difficult to detect SNPs and also achieve repeatable results. A more robust, repeatable, and accurate technology is needed to detect SNPs. Oskmer et al. Proposed using a label-free microarray imaging technique called Spectral Reflectance Imaging Biosensor, or SRIB, which is the machine shown on this slide. SRIB measures the change in optical thickness of mass on a glass slide, and this provides quantitative information of the binding interactions between the sample and the immobilized probes. Thus, it is able to quantify the hybridization of surface immobilized DNA with target DNA. They believe that this approach will help be able to distinguish single mismatch between the DNA fragments from a perfectly hybridized set of DNA fragments. Figures A and B above represent the before and after images of DNA microarray undergoing hybridization. There are 10 replicates of 9 different DNA fragments replicated across 4 arrays. In total, there are 360 DNA spots. The different replicates are labeled in Figure A. PM represents perfect match, MM represents single match, and DM stands for double mismatch. The numbers in front represent the length of the DNA strand, 20 for 20 oligomers and 18 for 18 oligomers. In Figure B, we see the after effects of hybridization, where brighter spots represent brighter hybridization and darker spots represent weak hybridization. Figure C quantitatively displays the before and after mass densities of the spots measured by the SRIB. Figure D represents the hybridization efficiency by plotting the percent of mass change upon hybridization. Perfectly matched DNA fragments are expected to see a 100% mass change, and this can be seen in the above figure for 20 and 18 perfectly matched fragments. The double mismatch samples were significantly different than the perfectly matched. However, the single mismatched fragment hybridization was not significantly different. So the researchers turned to kinetic denaturation to help distinguish between the single mismatched and the perfectly matched samples. The microarray mentioned in the previous slide was placed in a flow chamber to undergo kinetic denaturation. The idea behind this technique is that as the concentration of a salt buffer used to wash the microarrays decreased, the shielding of charges on the negatively charged DNA backbone will be reduced, destabling the duplex, resulting in denaturization of the DNA strands. Figure A on the left shows how the different DNA fragments change in mass density over time after they are washed with salt buffers. The DM samples decrease in mass density the quickest, while the PM samples take longer to decrease. The least stable duplex, 18 double mismatched, start denaturing when the microarray was washed with 2.5 millimolar salt buffer, while the 20 perfectly matched and 18 perfectly matched samples only start denaturing after 0.625 millimolar salt buffer is applied to the microarray, indicating they are more stable duplexes. The total mass density drop, however, is not indicative of the mismatches because each fragment starts with a different mass density. Thus, the figure on the right was generated to compare the decrease in mass density with respect to the initial mass for the 20 mer samples. From this figure, it is evident that the perfectly matched samples decreased the least, then single mismatched samples, followed by double mismatched samples, which denatured the most. The decay of this denaturation process is very similar to an exponential decay. Thus, the researchers believe that the coefficients used to fit the curves above could also be analyzed and used to assess hybridization of different DNA fragments. The exponential function that was used to fit the curves shown on the previous slide was a standard form shown above on this slide. The most critical coefficient is A2, as it is the decay constant. In the figure A above, the decay constant for the 20 perfectly matched samples and the 20 single mismatched samples are plotted and compared. There is a significant difference between the decay constants of the two samples. The same information was plotted in figure B for the 18 PM and the 18 MM samples. Again, there is a significant difference in the values of the decay constant. In figure C, the decay constant values of the two perfectly matched samples 18 and 20 were plotted, and there is no difference between their coefficient values. Finally, in figure D, the decay constant value of the samples, which is a positive hybridization, or the decay constant values of the 20 perfectly matched samples is found to not be different from the 20 DS samples, which is a positive hybridization control group. From this method, it is possible to distinguish the single mismatched samples from the perfectly mismatched samples, which was something the researchers were not able to do from the mass density loss alone. In conclusion, the new technology introduced in this paper has the potential to be a more simpler, more accurate, and more sensitive approach to detecting DNA fragments, with SNPs superseding the capabilities of mass spectrometry and fluorescence tagging. SRIB is a label-free detection method that can identify and discriminate the quality of hybridization reactions with the help of kinetic denaturation analysis as well. It is also a more cost-effective technology compared to current methodologies. Some future directions that could be taken with this technology include experimenting with longer stranded DNA and potentially indicating some way of knowing where in the DNA strand the single or double mismatch occurs. Thank you for listening to my talk.